Excellent. Good to see you, Yuli. Thank you for inviting me to this. We love telling our story, and it's been fun hanging out with you for the last, I think it's 2019 when we first met at JP Morgan. So
Yes.
you'll get an update on our story. So should I get started?
Yes, and yes, full screen mode, please.
Let's see if I can get this to work. Okay. Hi, everybody. I'm John Butler. I'm the CEO of QuantumCyte, and we're a technology company. And if you take anything home from this talk, it's precision and scale, right? So our company bridges the precision gap to deliver targeted cancer therapies soon. So how do we do that? What we do is we connect AI pathology to molecular testing, and I'll show you how that works. And the whole goal here is we want to give oncologists and patients the ability to make faster, more confident decisions about personalized treatment. Here's the agenda. To save time, I won't go through that. But I want to start off with the team again. I'm the CEO and CSO of the company. My co-founder is Dr. Vedant Chaudhuri. I've been developing genetic and genomics technologies for the last several decades. Badon has a PhD in MEMS, and this comes from the high tech industry. I've been working with Badon for about 15 or 16 years. We met at Pacific Biosciences, and the product that we developed, is really a high tech solution, both in the software, hardware, as well as some of the chemistry. Sarika Karudaparam has a PhD in biology. She's our product manager. She's been working at various biotech and medtech companies the last couple of decades. Katie Konigsfeld is our scientific product manager, who really runs the ship and works with customers. And we're super excited to be bringing on Carla Leibowitz, our chief business officer. Carla has been at Bain, she's been at Roche, she's worked at Page, digital pathology companies, and we just brought her on this week. And so we're super excited to have her. And then on the business and commercial advisory side, Dr. Paul Waring is an MD PhD. He was at AstraZeneca and Roche. He advises us on how to work with pharmaceutical companies and our pharmaceutical strategy, because pharma companies are our customers. Jake Doft is really more on the finance side. He invests heavily into pathology companies like Proscia and Page, and is very passionate about healthcare. And you'll see how we fit into this whole pathology workflow. And so he brings in not just the financial capital, but also the ability to work with very large organizations like Roche and Merck and put deals together. Donnie Rizzo is at Quest Diagnostics. He led their pathology and oncology business for about 7 or 8 years. He's now an advisor, and again, is advising us on commercial strategy as well as introductions to the Tempests, the Quests, the large pharmaceutical companies. And Achin Bhowmick on the high tech side is business strategy as well as high tech, working with some of the high tech companies. And our technical advisory team, Gene Davison on the computational side, Dr. Rich Pai is an MD PhD pathologist from Mayo. Ron Davis, I was in his lab for several years at Stanford. Anders Grant Jepson and Chris Love on the engineering side. So we have a pretty power packed group. So I'm going to read this slide to you because this sort of gives you an idea of what the technology does. So what do? We are the only platform that can precisely isolate tumor cells from a biopsy before testing. This allows us to detect the true genetic signal called biomarkers, which are like clues or fingerprints that help doctors choose the right treatment. So what does this do? It provides clean and precise view of the tumor. That way doctors can find the right treatment faster. They can help patients get access to new therapies earlier and even unlock options that would be visible using today's low precision medicine precision methods. So up in the upper right-hand corner, this is how the current process for isolating tumor works. The tumor is the dark purple area, and they basically go in from a biopsy and extract all of the tissue into a tube. So now you have a contaminated sample. What we go in do is we can go in and isolate DNA, RNA, and protein just from the tumor, so that when you run a clinical test, you're just looking at the tumor information. And this is a slide that Roche Diagnostics put together for us. We actually work very closely with them, but we bridge this gap between AI-based digital pathology on the left-hand side and next generation sequencing on the right-hand side. Very, very high precision methods for analyzing tissue samples, but we really bridge the gap because there is a gap that between these two workflows that where you lose all the precision in these workflows. So how does the technology work? So we start at number one. We take the input from an AI-based model that identifies where the tumor is in a standard biopsy. That's the green area. We then convert that. We take that input and put it into our workflow, where steps 2, 3, and 4, we create a image of that digital image, where the black areas are the areas we do not want to extract nucleic acid from, and the white areas are the areas that we want to extract nucleic acid from. And through our process, we're able to go and extract, again, tumor DNA just from the regions of interest, just from the tumor. And then what we do is we deliver a tube of DNA to downstream molecular processing. So we're a sample prep workflow. It's scalable, and we work with all of the digital pathology companies. So again, we're not a digital pathology company, and we've worked with companies like Navify, which is Roche, like a Halo Path AI, to integrate their workflows into our workflow, which is in the middle. So we're agnostic to the input. We're also agnostic to the next generation sequencing output. So we've worked with Illumina, Thermo Promega to provide this entire workflow. The product includes our proprietary software, an inkjet printer, our hardware, as well as consumables. We make money on the consumables. Our hardware is a full production unit. It comes with an 8-week lead time, full service and maintenance, out of the box, training ready to go at the lab. We also have the capacity to manage the throughput requirements for large reference labs like Quest Diagnostics and Mayo Clinic that are running about 100 samples per day. When we look at the competitive landscape, because there are other technologies that can do this tissue extraction procedure, none of them can provide high capacity and high precision, high sensitivity, and high throughput. Laser capture microdissection, some of you may remember this from grad school. It's a very high precision technology. It's probably the state of the art technology for doing precision, but it just doesn't scale in a lab. In the labs we're working with, again, are the Mayo Clinics, the Quest Diagnostics, Merck, Roche, AstraZeneca, that are doing large numbers of samples. Laser captured microdissection can maybe do 2 samples per shift, whereas we can do 80 samples per shift. So we're about a 10 or 20-fold increase in the throughput. And there are some other high throughput technologies like Xiol, as well as this manual process, that have high throughput, but they have low precision. We're the only technology in the market that can provide the level of precision and throughput. So let's talk a little bit about some of the data. This is a representative sample, and this is the sample. I love this sample. We have several examples of this in some of the clinical studies we've done. This is a patient that had colon adenocarcinoma, and we did a side-by-side comparison between our workflow and the current workflow. And this is a work we did at Singapore General Hospital with Dr. Tony Lim. In the yellow area in the upper left-hand corner, this is the result of the clinical test that they ran, and they were unable to identify any mutations in the panel for this particular sample. And you can see the size of the tumor is very, very small. When they ran the sample in our workflow, again, side-by-side comparison with the same sample, you see that we were able to identify 2 mutations. One is a P53 mutation and one is APC mutation. And if you look at the number VAF, which is called the variant allele frequency, which is essentially the sensitivity or the ability to detect these mutations, our variant allele frequencies are up 50 or 60% with these 2 particular these 2 mutations, showing that there's a very high percentage of the data that's showing that these it's giving a high confidence in the clinical call. This particular patient, actually, if they were using the current process, would not get access to an immunotherapy, cetuximab. But when they ran the sample in our process, they actually did get access to that immunotherapy. And we have several examples of how our technology is providing patients with therapies that they wouldn't otherwise get. In another study we did at Mayo Clinic, again, side-by-side comparison between us and the current process, we were able to pick up the same 2 mutations, as you can see, this PIK3A and BRAF, but we were also able to pick up 2 other mutations that would have indicated this patient could get access to another therapy. So we're getting patients more options for their therapy, and again, we have several examples of this in the clinical studies we've done. On our market traction, we have a lot of traction, and we've done about 7 digital pathology integrations. We have 7 active commercial deals with large pharmas and large reference labs, and we have about 26 issued and pending patents currently, and you can see some of the other information. We're really a tool, a platform that can be used for many different market segments, including biomarker discovery. We're a fully automated system. We bring automation to these reference labs. And we also see a big market in the area of proteomics, which is mass spec-based proteomics, which is for clinical trials with pharmaceutical companies. This is an example of our patents. So we have global patents. Again, we have 18 issued, 18 pending, sorry, 8 issued and 8 pending across the globe. And if we look at the revenue forecasted market share, we predict about 5% market share this is for the clinical testing market in 2030 with about $250 million of revenue with a pretty large TAM. We've raised $6.2 million to date. You can see some of the accomplishments. We're currently raising a $2 million round on a $10 million cap. On a safe note, we raised about $1.5 million to date, so there's about a $500,000 left. And we're raising this money to get cash in the bank as we move to close 2 of 3 deals, and we'll think we'll close 3 deals, 2 pharma and 1 with a large diagnostics company by the end of Q4. Once we close those deals, we'll definitely be Series A ready, where we're looking to raise about $7 to $8 million on about a $40 to $50 million valuation. Thank you for your time.
All right. Let's bring the judges onto the screen. And who wants to go first? You can raise your hand or just unmute yourself and ask. Faye, do you want to go? Do you have a question, comment?
I was interested too. Congratulations on all your progress to date, and you're working on integration. What have you have found to be the challenges, given like the complexity of the market?
Yeah, so we the technical challenges were pretty high. We went into this idea again 2019, and the logic was there's digital pathology, molecular testing, and we want to sort of bridge that gap, but we had to wait for the digital pathology market to catch up to our idea and even the molecular testing. So we've sort of shown technical validation. On the business side, it's really navigating our customers. Our customers are Roche, Merck, AstraZeneca, Mayo Clinic, Quest Diagnostics. Stanford's too small for us. They don't have the proper digital pathology infrastructure in place, right? And so it's been navigating those large organizations and finding the right stakeholder in those organizations. So that's been the biggest challenge. And then again, also working with these large organizations to get traction. So we're working very closely with Roche Diagnostics. We're about a year and a half into that collaboration, and now we're getting close to a deal. Working with Merck, same thing, 2 years to get that first inflection point. And for us, because we're a new product, because we're the only product that does this, and we've had to sort of educate the industry, we need those two. We need one of those deals, just one, and that validates. And that's coming from the Series A VCs. They say, look, John, we love what you're doing. We love the product. We get it, but you got to show that somebody will actually buy it. Now, we do have revenue from sort of a services thing where we do proof of concept and we get data for internal marketing within the organizations, but we need one of those 3 deals, just one of those 3 deals to demonstrate, I guess, product validation. We're close, and we're very confident we'll get those deals, but that's the biggest challenge. I don't think we really understood that back in 2019, 2020 when we did this pivot. So we're getting better.
Yeah. It's a tough business, man. But you guys are sticking with it, and so that's good. Martina or Laurie, you have unmuted.
She asked the question I was going to ask.
Yes.
Okay, Martina.
I can tell you it's a very, very cool product. If anybody wants to do a virtual demo, we'd love to show you the stuff. Happy to do that. But it's our customers are the largest diagnostics company in the world. And I'm sure everybody's had experience working with, maybe not Roche, but we do have experience. If anybody's interested in having coffee and talking about sort of how we navigated pharma and Mayo, happy to have that conversation. But yeah, so we're at that point where we've been sticking it out, and now we're working with these organizations. They love the data. We had to bring in the Donnie Rizzos, the Carla Leibowitz of the world to get those deals. My co-founder and I don't close deals. We need people that could actually close them. So we're excited to have brought in people that have actually closed deals and done M&As with Roche and so forth. So it's an exciting time.